All right, everyone. Thank you so much for joining us today for another Suds and Science talk. My name is Andrea Decker, and I have the pleasure of representing the Beat Science Center in Balboa Park. That's this beautiful building in the background. In case we have some first-timers here, welcome. We're really excited to have you here. Everybody that is regular, so good to read your names on the screen, and thank you so much for joining us yet again for another talk. So before we get started, a couple of things I would like to explain, especially for people that haven't joined us for Suds and Science yet. Suds and Science is not your typical lecture series, so there's not going to be a huge PowerPoint presentation and a long talk by a presenter. We mainly want to keep it conversational, so our speaker will give more of a verbal introduction to his work with just a couple of PowerPoints and a little video. And then the majority of the evening will actually be spent talking to each other, asking questions. Jorge has a couple of questions for you too, so I'm excited to see what discussion items will be brought up tonight. So the beginning part, as you notice, I hit the recording button. So we're recording the beginning part because we would still like to be able to share Jorge's work with a wider audience. So this will be on our YouTube page in about 2 weeks or so. But once Jorge is done with his presentation and we get to the more conversation part, I will turn off the recording. I'll give a quick official goodbye to our audience at the end for our video audience on YouTube. And then I'll stop the recording, and then hopefully we will have some of our audience feeling comfortable enough to turn on their camera, because it will just be us. And that'll, you know, when we have the camera on, it just makes for an easier engagement with each other. But you don't have to, it's just a suggestion so that we can all engage with each other a little bit better. As you might know, Suds and Science is normally a program that takes place in bars and restaurants around San Diego. We have 4 neighborhoods that we host this in on a rotating basis. 1 month we're in North County, then we're in South County, then the next month we're in Central San Diego, and then we are in East County. But of course that unfortunately is not possible, and our beautiful building continues to be closed at this time. But science never stops, whether we are closed or not, which is why we are very grateful for the virtual world that we are still able to bring these programs to you and still help you enjoy some science, learn some science, and engage with some of our amazing scientists here in San Diego and engineers. Okay, and I think that kind of sums it all up. Yeah, I think that's about it. Jorge Urresti is our speaker for tonight, and I'm going to let him introduce himself. So Jorge, why don't you take it away?
Thank you very much, Andrea, for everything. And thank you very much, everyone, also for coming here and spending this evening with me and Andrea. Hopefully it's profitable and you learn a lot. And well, my name is Jorge Urresti. I am from Spain. I came to the US around 6 years ago to study autism genetics. And I started my scientific career back in Spain, specifically in Barcelona. I did my PhD working with cancers or tumors of the brain system, of the nervous system. So I was interested since I was very young in doing scientific work related to neuroscience. So when I had the opportunity to move to the US and I started working on neurodevelopmental disorders, more specifically on autism or autism spectrum disorders, as they are called now. So then I was very eager to do that. So when I moved here 6 years ago, I started doing my postdoc at the UCSD in the lab of Lilia Yakucheva. She's a geneticist, and then we study autism genetics. And then, so there are 2 different, today what I'm going to explain kind of 2 different things. So I'm going to explain how the field of autism genetics has advanced a lot in the last 10 years or around 20 years. The different discoveries, like the big picture, and that has been discovered because a lot of things have been explained now. I mean, 20 years ago, we didn't know a lot of the things that we know a lot now about autism genetics. No, what mostly that is a genetic disease, and most of the different genetic variants that are high risk for this disorder. And then after we identify these genetic variants, then we need to know how they are actually working, no? What they are actually doing at the physiological level, no? What they are doing is impacting the development of the brain. So we need to know actually what is the impact that these genetic variants are doing in order to understand better this disorder or this disease, and then to be able to discover or to perform new therapies or new treatments, or for, well, it's always for the, as sorry, as you better understand something, I believe that you have a better opportunity to help the people, no, that is affected by it, no? So that's the main drive behind all the research that I have been doing during all these years. So yeah, and then, well, actually, and then I moved. Actually, I moved this month. I started working for a private company that we are also doing stem cell research related to neurodevelopmental disorders. So yeah. So before I actually start going to the talk in itself, I actually mentioned it 2 or 3 times now. I will use the term genetic variants, okay, or genetic mutations, quite a lot, okay? I kind of assume that everyone knows what I am talking about. If somebody doesn't know, maybe you can say something on the chat or share your hand, and I can just explain it very quick before we start going in. What is genetic variation? No, if I say genetic variation, you understand what I'm talking about. No, well, I'm just going to explain it very fast. It's just 2 minutes. So as well as I guess most people know, no, but I guess you know, in our cells we have DNA, which is the basis of the proteins that form our body, no, the cells that form our body. And then most of these proteins have a sequence that is written in the DNA, and this sequence has a consensus. I mean, it cannot deviate too much in order for the protein to perform the function that it has to do. If there is some mutation, some changes in this sequence, this can lead to that the proteins that are going to lead, so they are not going to be functional at the correct level, and this may lead to disease, no? I mean, there are plenty of examples of different diseases, no? Or cancer, for example, is normally because of mutations in the DNA, no? So this is when I am talking about genetic variants, I'm talking about this, no? About differences in the DNA when compared to the consensus sequence that is called, okay? So, well, so autism, as I guess everyone knows, no, has been a pretty controversial disorder for a while. There is a lot of different theories going around and a lot of different histories and urban theories and conspiracy theories about it. So, well, I'm not going to talk about it, at least right now. Maybe on the Q&A, if you want, we can talk a little bit about it. But, well, the thing is that in the last 10 years, well, it was already suspected or kind of known that autism was a genetic disorder. No, because there, for the people that was studying, the psychiatrists and psychologists back in the 90s or early 2000s, I mean, just by looking at the prevalence in the different kinds of population that has autism, you can see that there is a big difference between the general population, between siblings, or between siblings that are twins that come from the same zygote, or that doesn't come from the same zygote. No? So I'm going to show very, very quick just one graph. So it's going to be easier for you guys to understand what I'm talking now. So here, as you can see, okay, so in the case of monozygotic twins, which means that are siblings twins that are formed from the same zygote, so this means that they have the same genetic background, okay, and they are completely identical, the prevalence of autism is around the 70 to 90%. No? So if one of the siblings is diagnosed with autism, it's very, very likely that the other sibling is also going to be diagnosed with autism. In another way, in the case of dizygotic twins, no, or twins that they are also, that they share the womb, but they don't have the same zygote, so they don't necessarily have the same genetic background. Well, actually they don't. Then this prevalence goes down a lot, okay? It goes around 10 to 20%, which is actually pretty similar to regular siblings, okay? And then the prevalence in the general population is obviously lower. It's, well, actually this is outdated now. This is a little bit higher now, which is, I'm going to come by later too. But well, since just by these observations, even if at the moment that this was studied, this is on the 90s or early 2000s, we didn't have the means to understand what was going on at the genetic level, no? So we already knew that something was going on, no? That this disease, it was probably caused by something related to the genes, no? To how we are developing. But then something happened specifically at the early 2000s, but more than in 2010s and later on, that give us scientists or humanity in general tools in order to understand better what was going on. I mean, well, I don't know if anyone knows what I'm talking about. I mean, I guess it's very, I don't know. I wanted to make like a quiz or something if someone wants to guess what I am talking. But I mean, it was a pretty big discovery. I mean, actually, I think that the first person that did the first was here in San Diego. And I am talking about the genomic sequencing. So the first genome sequence that, if I remember correctly, was Craig Venter. Craig Venter now has I think it has a company and also has a scientific team here in San Diego. So, well, so yeah. So the thing is that with the advent of genomic sequencing, so we were able to actually read the genomic code. No, we can now look at the genome of any person. No, you can just give a blood sample or any sample, any tissue sample, and they can look at all the whole genome or the 21 chromosomes. So this obviously gave us a whole lot of information in order to be able to start to study the differences between the patients and the healthy, the regular people. No, so this is just also to exemplify. No, the cost of the genomic sequencing. It went down a lot, specifically around the 2007, and then right now it's very cheap compared to when it started. No, so as you can imagine, when these tools started to develop, and then the sequencing technologies were more available to the general hospitals and people. The number of people that were able to get tested and the number of samples that we could get in order to start studying this, so it was very big, and then is when we started to make these discoveries. So how we actually study how we discover genetic variants, no, between that we know that are going to be specific for autism, no, that we can say, okay, if a person is born with this specific genetic variant, it has a high risk to manifest autism spectrum disorder in this particular case. No, it's the same strategy for other diseases that are also neurodevelopment or for genetic diseases in general. So we do it with something that it's called the trio studies or family studies, which is also very simple if you think about it. So basically, we just take the DNA from the parents, okay? Obviously, if we have more information on the family tree, if we have the grandparents or if we have even siblings, obviously it's even better. The more information, the better. But this is just the most simple case. If we just have DNA from the parents and then we compare it with the DNA from the sons, then we can see, I mean, as when you have offspring, no, the offspring of the parents is not 50% dependent on the mother. No, I mean, everything is hereditary, but there is always a percentage that is new for the world, for the new person. No, and then we will see that is specifically in this part, in this de novo variation, which is the most high risk for the autism. So yeah, basically this is what we do. Okay, so we take the DNA from the parents and from other people of the family, and then we can compare it with the DNA from people with autism, and then we can identify these genetic variants. Okay, so I don't really want to go very deep into the number of genetic variants and everything. It's very technical, and there is a lot. That's another thing but I want to say that, I mean, there's a lot of another thing that has been shown thanks to this research is that autism has a lot of heterogeneity. As if you know anyone with autism, or if you are involved with any foundation that works with families or something like that, you probably notice that there is no two single people with autism that is the same. Okay, there is the number of different symptoms, the number of the degree of which the symptoms they manifest can vary a lot. Okay, so there is a lot of, I mean, this is why it's so difficult also to diagnose. No, I mean, there is a lot of and I think this is also where this controversy during all this time comes from. No, that it was difficult. I mean, there is the obvious cases, very extreme cases that you can see, but there is also these very milder cases or something like that that are tricky to diagnose and to treat. So there is a lot of a spectrum. This is why the definition was changed. No, it's not autism now, but it's called Autism Spectrum Disorders. So this is the same in the identification of genetic variants. Okay, when we started, now there is like thousands and thousands and thousands of families, no, that have gone through this kind of research, and then we have been able to study the genetic variants of a lot of different families, and then it has been discovered that the number of different genes that are risk for autism is very, very, very big. Okay, I don't know. For example, there are diseases. I don't know, cystic fibrosis, for example, no, or cystic fibrosis is I guess it's a disease that is caused by one gene. No, so if there is this gene, and if you have a mutation in this gene, you are going to develop cystic fibrosis. For autism, there is hundreds of genes. there is hundreds of genes, okay? There is, and it doesn't necessarily mean if you're going to have one mutation in one of these genes, doesn't necessarily mean that you're going to develop autism. It's still something that we don't know. This is called the penetrance, okay? This means that even if you have these mutations, there are still other factors that they are still not very well known, that it doesn't necessarily mean that you're going to manifest this disease. And then finally, another thing that I want to say is that a lot of times when we talk about genetics disorders, we think that is hereditary, no? That something that is genetic, it comes from your parents. But that is not necessarily the case, okay? Most of the times, actually, that is not the case. So that something is genetic, it means that it's in your genes. But as I mentioned before, there is a lot of your genetic background that doesn't actually come from your parents, that it comes new for you. And most of the autism variation has been discovered that is actually in this portion of the back of the genome. Okay, so because I know that is sometimes there is a little bit of parent blaming, no, or even if you have a son and your son has autism, sometimes there is a little bit of self-blaming, no, and stuff. So I want to take that out of the way, because I mean it's never anyone's fault. Obviously, it's not like you can control it anyway, no, obviously, but even if that was the case, okay, it has been discovered that in the big percentage of the cases, the genetic variants that are identified in the sons, they are new, they are not present neither in the parents. And then most of the mutations that are identified for autism, they affect the brain, okay, as you probably imagine, okay? They are related to the brain function, specifically to the brain development. So the impact that these mutations have starts manifesting during the fetus stage, during the pregnancy, even before you are born. Okay, and then later on, when you were born, obviously, I mean, well, humans, we are still not fully developed now when we are born. So, I mean, the thing is that this kind of mutations, they impact the development of the brain in a way that later on, when you are adult, you manifest these symptoms of autism, no? So this is where I want to go. There are two different things, no? One thing is to identify these variants, no, which I explain now very briefly and roughly how we do it. And there are the other thing, this is how we know what these variants are actually doing to the brain, no? These are two different things, no? And in order to do so, the strategy, I mean, there are several ways to do it, no? But lately also, during the last decade or something, there is one strategy that has gained track because it has a lot of advantage compared to the other strategies, which is the use of stem cell research. Okay, what are your opinions on the stem cell research? Is anyone here that has strong opinions on that matter? I mean, is like, do you think that it should be allowed, should not be allowed? I mean, well, I guess that here, everyone in California, I mean, it's very...
I know that we had some reactions to stem cell research talks before, where people were feeling hesitant about it because they didn't quite, there were too many conflicting reports in the news of what it means, where stem cells come from and all that good stuff. So
yeah, actually, you guys here in California, you had to vote for a proposition in the last elections, if I remember correctly.
Actually, one of our guests just said it was on the ballot this year.
Exactly.
And she needed more information to really demystify the topic, because there was too much different and diverse information out there that I think made it more confusing for people. And we have someone here who said, yeah, that should be allowed, stem cell research.
Yeah, so the thing about the kind of stem cell research that we do, I'm going to talk first about why we do it. No, the model. So as I explained before, no, the autism as well as other disorders like schizophrenia, no, or intellectual disability, all these kind of neurological, neurodevelopmental disorders, we already know that they are genetic disorders and they are from the development. So they start at the womb. In order to study that, how do you do it? I mean, you cannot just take samples from fetuses. That's pretty obvious, no? You can work with abortions, no? I mean, that's also controversial, kind of, okay? You can also work with material from abortions and stuff, but obviously this is very limited. It has ethical implications, I mean, which I guess, I mean, it's obviously worth discussing in another time, no? So it's there, no? We can also study, I mean, you can also use to study post-mortem brains. Okay, I mean, you can also, I mean, there is people with autism or other disorders and they get into adulthood or or no. And then when they die, you can just take a sample from the brain and then you can study that. But obviously that also has obvious limitations because you are looking at the stage of the death, okay? And what we want to see is the stage of development, no? So you are not going to, you are looking at the stage that is already, what you wanted to see what is happening has already passed, no? You are looking at the consequences, no? But you're not looking at what is causing that, no? And then you can also use animal models, no? Which mice is probably the most straightforward and the most used, no? But then that's another problem because in the case of these kinds of disorders, no, neurological disorders, the brain of the mice is totally different from the brain of the humans, no? Mice are very good to study toxicology or to study things more related to the metabolism because in that way we are not that different. But when you want to study something that is related to the brain and higher cognition functions, then obviously mice start having obvious problems that they don't really translate very well the findings that you're going to have in mice to the findings in humans as is natural. And then using other animals, no, as monkeys or something like that, obviously can be done, but this is not something that I personally advocate. And I think that it also has, I mean, obvious problems, not only ethical concerns that I think they are pretty obvious, but also, I mean, the difficulty to have to work with this kind of animals, no, which I guess is going to be pretty big too. Therefore, what is the other model that we can use? Okay, so here it comes stem cells. No, stem cells, as I guess that most people know, they are the cells that can differentiate into different cells of the body. No, I mean our body has different kinds of cells. No, the cells of the eye are different from the cells of the skin, or from the hair, or from your heart, or for different parts of the body. They are different kinds of cells that they are designed to do the function depending on the organ that they are. But then stem cells, they can, I mean, stem cells, they are like naive cells. They don't, their function is to proliferate, to double, and then to differentiate into the cell type that they want to do, that they need to differentiate at the specific moment. No, I mean, this is a very brief definition, okay, because now we know that the only real stem cells are the embryonic stem cells, okay, that they are the ones that are in the embryo, that they really have the potential to differentiate into all the other cells in the body. But then when we are adults, we don't really have real stem cells that can differentiate into everything, okay? We have more specialized stem cells for every organ. We have neural stem cells that can differentiate into neurons. We have cardiac stem cells that can differentiate. I mean, so we have more specialized ones, okay? But this is just information. Don't really want to delve into that. So what we can do, okay, so a big discovery that it was also in the last decade, okay, is that what we can do now, we can take samples, biopsies from the skin or from other parts of the body, okay, normally it's the skin because it's just very simple to take a biopsy from the skin. You can take the cells from your skin, you can culture them in the lab, and then you can do some things, okay, add some factors and experiment with them, and you can reprogram them to turn them into stem cells. Okay, and then you can have a pool of stem cells of a given individual, and then you can actually differentiate these stem cells into whatever you want in the lab. Okay, so given this technology, okay, so now you can take cells from an individual, you can grow neurons or astrocytes, okay, or neural cells in the lab, and then you can study how they behave compared to cells from other individuals. No, so in a pinch, that's kind of what we do. Okay, this is basically what has been my research. Okay, I just take cells from patients with autism. I take cells from healthy people. We differentiate them into neurons in the lab, and then we can see how these neurons behave. Okay, I mean, neurons normally they communicate with each other, so you can investigate the extent of this communication. If they are communicating properly, they normally they have to migrate into different layers of the brain, so you can also look at these potentials. No, and then you can infer, no, the things that I mean, the impact that this is going to have in the brains of the people with autism, and then on the basis of that, you can start wondering. So the main model that we use actually, okay, is a little bit more complicated. It's just not just neurons. Now we can do one thing that is called brain organoids, which is a kind of 3D models of like masses of cells, okay, that they self-organize into each other, and then they are very, very similar to the early stages of brain development. So they mimic very well these early stages of the fetus that are the ones that we know that is when these kind of diseases manifest. So they are a very good model in order to study this kind of the neurodevelopmental disorders. I actually have a very small 2-minute video, okay, that is actually, exactly, they are also called mini brains. Yeah, different names. I actually have a video that is also from people from here, from San Diego. They are actually my colleagues, that in just 2 minutes, they explain very briefly what they are brain organoids, and they also show them. I mean, how they are. So I'm just going, it's just 2 minutes. So I guess you guys can see it. Let me share the screen.
When you share that, make sure that you share audio too. There's a little button that says share audio, computer audio.
It's okay? Can you hear it?
Yeah, we can see it, but not hear it. Is there supposed to be?
Oh, my bad.
When you stop sharing your screen, Jorge, for a second.
Sorry.
And then when you click on share screen, on the left lower corner, there's a little button that says share computer sound.
Okay, my bad.
You wanna, yeah, you wanna click that. And then hopefully it'll, yeah, now we hear it.
Oh, sorry.
These researchers are using clusters of neural cells called brain organoids because they have many of the characteristics of a developing brain.
These are brain organoids. We call them brain organoids because they develop similarly as a human brain. We don't make these organoids by putting cells together. We just give cells the right conditions to grow, and they organize themselves into these structures. Like many stem cell researchers, we first start with induced pluripotent stem cells. These are made from a mature cell, such as a skin cell. The skin cells are given different factors, and they become pluripotent stem cells.
Okay, so
To
this is what I
generate
explained
brain
before.
cells, we provide specific growth factors to guide the cells to become neuroprogenitors. These cells are the first step in brain development, and you can create all types of brain cells. First, these cells self-organize into neural rosettes, and we keep providing them with nutrients and factors for them to become healthy, mature neurons. As these rosettes grow larger, the cells form different layers that are similar to the ones found in the brain cortex.
These brain organoids are the focus of these researchers' many investigations.
One focus is to grow brain organoids from pluripotent stem cells, from individuals who have neurological disorders. These let us see what differences there are between normal and abnormal development, and even ways to treat the disorder. Brain organoids also let us see very early brain development. These earliest stages of brain development are when errors that cause disorders occur. Disorders that will affect the brain for life. And this is why we're working with brain organoids. To reveal how a brain is built, so we can see how it works, how neurodevelopment can go wrong, and how we can prevent that or fix it when it does.
Okay, so I mean that's basically what I was explaining, no? Maybe better. So yeah, so that's the main focus. That's basically the main focus of the research that I have been doing. Okay, I have been using brain organoids in order to study autism spectrum disorders. Okay, in my specific case, what we found, okay, I mean because it also has been found in some cases of autism in the postmortem brains when they look at the postmortem brains of adults. They can actually see that some of the layers that form the brain, they are a little bit disorganized. They are actually like anatomical differences, small. And then in our model of organoids, one of the things that we found that happens is that the neurons, they don't migrate properly. So, I mean, in these models of organoids, obviously it's not the same as the brain. You don't see the 7 layers that form, but you can actually see up to 3-4 layers, depending on the success that you have growing these organoids. And then you can actually, I mean, you can quantify the extent of how the neurons are migrating in these layers, and we can even make some cute videos. And then, so we basically observe that for one specific mutation of autism, that is actually the one most common, the neurons don't migrate properly. And this is probably one of the causes that they have these differences. No? And then just to finish, no, because I think that I am going to go overboard in a few minutes. I also wanted to mention, I mean, I have been more focused obviously on brain organoids because this is the focus of if you want to study neurodevelopmental disorders, obviously. But then there is also not only brain organoids, there are organoids for all of the organs of the body at this point. Okay, they are organoids for the intestine, they are organoids for the stomach, they are organoids for the I don't know that for los riñones, for the kidneys, sorry. This is the Spanish sometimes. So yeah, and the thing is that these organoids, okay, they are actually more advanced. The field of this kind of organoids is more advanced than the brain organoids, and they can be used also to, I mean, they are now being studied to be used for transplants, for example, okay? You can, I mean, not the whole organ, but you can maybe grow pieces or small organoids of the kidney or of the intestine. This is not with the intestine specifically, but I am sure that in the future we're going to see it with other organs. You can grow small pieces of the intestine in the lab, okay, and then you can actually use them to patch people that has tumors or periodontitis or other things, no? And then also for tumors, you can also grow organoids of tumors, okay? You can take biopsies from tumors from patients, you can grow organoids in the lab, and then one of the most widely used for a lot of different tumors is the chemotherapy. That is a cocktail of different drugs, no? Normally for each different type of cancer, the combination for every person, this combination of drugs sometimes, well, depending on the cancer and everything, has to vary, no? I mean, it has to be different. And finding the right combination and the right concentration of these drugs for that specific patient, most of the time is just a matter of trial and error. No, you need to try different dosage and see how they behave, how they react, and then just change it accordingly. So if we use organoids, okay, we can actually take a biopsy from this patient, okay, we can grow organoids in the lab, and then we can treat these organoids with all these different combinations. And then we can do a screening of a lot of different combinations of drug in these organoids. We can find the right one, and then we can treat the patient with the correct combinations of drugs that that is going to work for him, without having to go with all this process of trial and error that obviously also has more side effects and everything. No? So with this, I just wanted to exemplify kind of that the many things that we can do with the stem cells. Okay, so they are not only useful for research, but they are also useful for tissue replacement. Okay, they can also be used for precision medicine, which is the example that I mentioned now, to discover the correct medicine or the correct dosage of drugs for a specific disease. I just put the example of cancer, but this can work for other disorders. And also in research for disease modeling, it's also a very good tool for researchers in order to study these disorders, because they allow us to mimic the organ that we want to study. We also have the same genetic background as the patient, which is also very important, that this is not something that you have if you use a mice model or something like that. So obviously it has these benefits that other models doesn't have. So hopefully I dispelled a little bit of the doubts that you may have with the stem cell research. Well, we still have time, so I am open to any kind of question that you guys have on this topic, or also on autism, because I know that I have been a little bit talking about two different topics kind of. So I don't know if you guys have any question or anything that you want me to maybe elaborate a little bit more on. I can also spend more time doing it.
Perfect. Thank you so much, Jorge, for leading us through your work and the discoveries and how you actually do your work. With that, I'm going to say goodbye to our YouTube audience. Bye, YouTube audience. I hope you're going to enjoy this talk, and I encourage you to join us live, because you're missing a good part of the event, which is the Q&A and the discussion and the conversation that we will have with Jorge now. So I encourage you to join us next time for the live event. It happens on every second Monday of the month. In the virtual world, we start at 7, and I think normally when we go to bars, we start at 6:30. So it's right about dinner time. We can all sit down in a restaurant and a bar, have a drink, have something to eat, and have a good conversation with each other. And with that, thank you, audience. We will see you soon.